Namaskar, welcome to PK's classes. Today we'll study mechanism of resistance to anti-amoebic drugs. So when we say anti-amoebic drugs, it acts against amoebiasis and the causative agent is Entamoeba histolytica. So this Entamoeba histolytica exists in two forms, cysts and trophozoites. The cyst is the infective form, and inside the body the trophozoite is the active form, whereas the cyst is the inactive form. And this cyst, which is the infective form, that through fecal oral route helps in transmission of Entamoeba histolytica. So the source of amoebiasis is contaminated food. And if you see, through contaminated food, the cyst is ingested, and in the small intestine, the cyst wall is weakened and releases the trophozoites. These are the trophozoites, and these trophozoites are labile and they live inside the body, are invasive, enter into the intestinal wall, can through blood circulation goes to the liver. In this way, the amoebiasis, we can say, is of two types: intestinal or luminal amoebiasis and extraintestinal, that is when it affects the intestinal wall and the liver. And the cyst is then these trophozoites, they burst to again release the cyst, and the cyst is expelled through the fecal matter, and the cyst can survive outside the body for several weeks. So through contaminated food, by flies, it again can infect another person. So as we said that the amoebiasis can be luminal in the intestinal lumen, so luminal or intestinal, or can be extraintestinal or tissue amoebiasis, where it can infect the intestinal wall and liver. Accordingly, we can have two types of drugs: luminal amoebicides or intestinal amoebicides, which includes the dihaloacetamides like diloxanide furet, clefamide, itofamide, teclosan, and the halogenated hydroxyquinolines, diiodohydroxyquinoline, that is iodoquinol, iodochlorohydroxyquinoline, that is cleoquinol, enteroquinol, antibiotics like tetracycline, paromycin, erythromycin, fumagillin. Then extraintestinal, mostly in the liver, extraintestinal amoebicide, that is chloroquine. Then both intestinal and extraintestinal amoebicide, that is nitroimidazoles, metronidazole, tinidazole, secnidazole, satronidazole, nimorazole, benzimidazole. These are the common group of drugs against amoebiasis. The alkaloids, imetin and its derivative dihydroimetin, and the new group of drugs, thiazolide derivative nitazoxanide. So the luminal amoebicides are mostly effective against the cysts, whereas the intestinal and extraintestinal amoebicide drugs, that means those which act in the liver and intestinal wall, they are effective against mostly the trophozoites. And this group of drug, this nitazoxanide, is effective against both cysts as well as trophozoites. So let us first discuss the mechanism of action of these drugs, then we will go for the mechanism of resistance to these drugs. So nitroimidazole group of drugs, the most common group of drugs, they are the prodrugs. After entry into the cell, they are activated by reduction of the nitro group. So there will be supply of one electron, and that electron is supplied by reduced ferredoxin. Reduced ferredoxin provides the electron, and the nitro group is reduced, and then that can generate the free radicals which can damage the DNA, nucleic acids, DNA, RNA, and can cause cell death, and they kill the trophozoites. And this reduced ferredoxin in the process is converted to ferredoxin, and again ferredoxin can be reduced by the enzyme pyruvate ferredoxin oxidoreductase. So for a reduction of the nitro group, we need reduced ferredoxin, which in the process is converted to ferredoxin, and for again the recovery, the ferredoxin is reduced by the enzyme pyruvate ferredoxin oxidoreductase. So we need ferredoxin as well as the enzyme pyruvate ferredoxin oxidoreductase for this. So ferredoxin is first reduced by pyruvate ferredoxin oxidoreductase, then that is used by the nitroimidazoles, and so this enzyme pyruvate ferredoxin oxidoreductase is the key enzyme in anaerobic metabolism, and this is present in the protozoa, the Entamoeba histolytica. Then the emetin and dehydroemetin, they mainly bind to the ribosome. They interfere attachment of tRNA to the ribosome, and then they inhibit the protein synthesis. Emetin in addition can produce also the reactive oxygen species, and they have direct lethal action on the trophozoites. Then nitazoxanide, it inhibits the enzyme, here I said pyruvate ferredoxin oxidoreductase. This is inhibited by nitazoxanide. And then the nitazoxanide is effective against both cysts as well as trophozoites, and it is acting against the means acting in the intestinal lumen as well as in the intestinal wall. Then we will go for the luminal amoebicides like diloxanide furoate, which inhibits protein synthesis by inhibiting the polymerization of amino acids to polypeptides. Iodoquinol, enteroquinol, they have the halogen group which they use to halogenate the proteins. They interfere with essential enzyme systems in the parasite, and they also form complex with metal ions vital for metabolic process of amoeba. In this way they kill cysts. These are luminal amoebicides. They kill the cysts but not the trophozoites. Chloroquine is the drug which is effective against the hepatic abscess. They act against the liver because they are highly concentrated in the liver, and they inhibit parasites' ability to utilize hemoglobin. So this is basically the antimalarial drug. So it destroys trophozoites but not effective against cysts. Among the antibiotics, paromycin inhibits the protein synthesis by binding to 30 subunit of ribosome. So now we will discuss the mechanism of resistance, and in this one figure we can discuss the mechanism of resistance. First mechanism, if there is reduced uptake of metronidazole, so metronidazole drug concentration will be less inside. the amoeba. Second, by reduced expression of either ferredoxin or the enzyme PFOR. Third, by increasing efflux by overexpression of P glycoproteins. So there can be efflux of the metronidazole. So that will reduce the concentration of metronidazole inside. Fourth, so the active metronidazole can oxidize the DNA and can damage the DNA. So there can be increased DNA repair activity. So that is a mechanism of resistance. There can also be increased antioxidant defense, so that the oxidation of DNA can be inhibited. There can be mutation in the iron transport protein which will affect the level of ferredoxin. There can be increased rhamnose metabolism which can alter the pyruvate metabolism and affect the level of PFOR. And also there can be alteration in the ribosome, which is the target. All the drugs affecting protein synthesis, this can be the eighth mechanism. So let us discuss one by one. So reduced uptake due to changes in membrane permeability, so that will decrease the intercellular drug concentration and can be a cause of resistance to metronidazole, imetin, dihydroimetin. There can be decreased expression of ferredoxin and pyruvate ferredoxin oxidoreductase enzyme, so that will decrease the reduction of nitroimidazole, so there will be fewer toxic free radicals, so that will decrease the drug efficacy and hence cause the resistance. There can be increased drug efflux from the parasite due to overexpression of P glycoproteins or ATPases, so that will decrease the intercellular drug concentration and hence can be a cause of resistance to drugs like metronidazole, imetin, dihydroimetin. There can be upregulation of DNA repair mechanisms, so enhanced repair of metronidazole induced DNA damage, that will reduce the cytotoxicity and hence decrease the efficacy and can be a cause of resistance. The fifth mechanism of resistance is increased antioxidant defense due to the overexpression of antioxidant defense mechanisms like the enzyme superoxide dismutase and peroxyreductase, so that they will protect the parasite from the nitroimidazole induced oxidative damage. Then the sixth mechanism is mutation in iron transport protein, and the iron containing protein is the ferredoxin. So mutation in iron transport will cause the deficiency of the ferrous iron transporter, and that will impair the activity of iron containing protein like ferredoxin, which is essential for the activation of nitroimidazoles. So there will be no activation of nitroimidazoles and hence decreased efficacy and hence resistance. Then there can be overexpression of the rhamnose catabolism regulator, which will alter the pyruvate metabolism and hence suppress the expression of the enzyme pyruvate ferredoxin oxidoreductase, and that will decrease the production of reactive oxygen species, decrease the oxidative stress, and hence decrease efficacy and cause resistance to metronidazole, particularly in certain bacteria, because metronidazole is also having antibacterial action. Finally, the altered ribosomal binding sites, so the drugs which bind to the ribosome like imetin, dihydroimetin, the antibiotic paromycin. So due to altered ribosomal binding site, there can be decreased drug binding affinity and hence resistance. So this is all about the mechanisms of resistance to anti-amoebic drugs. Thank you.